This is a primary photosensory neuron in the vertebrate's eye called as rod cell. When we will look into the upper part of rod cell, it will look like this. This is the plasma membrane of the rod cell. This is the disc membrane of the disc compartment. This disc membrane has a photoreceptor called as rhodopsin or RH. Whenever the light is absorbed by inactive rhodopsin, the excitation process of the rod cell starts. Due to this light absorption, the 11-cis retinal in the inactive rhodopsin is converted into all trans retinal. As a result, the inactive rhodopsin or RH becomes active. Another protein called as transducin hovers nearby on the cytosolic face of the disc membrane. Here, the transducin is denoted as capital T. The transducin has three subunits like T alpha, T beta, and T gamma. The activated rhodopsin then catalyzes the replacement of GDP by GTP on transducin. The transducin then dissociates into T alpha GTP and T beta gamma. The T alpha GTP then binds to the inactive PDE or cGMP phosphodiesterase. The T alpha GTP then removes the inhibitory subunit denoted as capital I of the inactive PDE. Due to this inhibitory subunit removal, the inactive PDE becomes active. The active site of active PDE is present on the cytosolic side of the disc membrane. The plasma membrane of rod cell has cGMP-gated cation channel in an opened state. Through these opened cGMP-gated cation channels, sodium ions and calcium ions pass into the cytosol of rod cell. The cation channel protein bound cGMP then binds to the active PDE. The active PDE then catalyzes a biochemical reaction in which the bound cGMP is converted into a biologically inactive compound 5'-GMP. The 5'-GMP then dissociates from the active PDE. Another cation channel bound cGMP is also converted into 5'-GMP in presence of active PDE. Due to this conversion of cGMPs to 5'-GMPs, the concentration of cGMP decreases. As a result, the cation channels become closed. Due to the closure of cation channels, the influx of sodium ions and calcium ions is prevented. Means the sodium ion and calcium ions cannot enter the cytosol of the rod cell through closed cation channels. Due to this prevention of influx of sodium ions and calcium ions through the cation channels into the cytosol of rod cell, the plasma membrane of the rod cell becomes hyperpolarized. This signal then passes to the brain. With this step, the excitation process of a rod cell ends. Now, let us discuss about the recovery or adaptation process of the same rod cell. The plasma membrane of the rod cell has sodium ion calcium ion exchanger. Through this sodium ion calcium ion exchanger, 1 calcium ion and 4 sodium ions are exchanged continuously. Due to this continued efflux of calcium ions through the sodium ion calcium ion exchanger, the cytosolic calcium ion concentration decreases. This decrease in cytosolic calcium ion concentration causes the conversion of inactive guanylyl cyclase or GC into the active guanylyl cyclase or GC. This decrease in cytosolic calcium ion concentration also inhibits the PDE. The cytosolic GTP then binds to the active GC or guanylyl cyclase. The active GC then catalyzes a biochemical reaction in which the bound GTP is converted into cGMP. The cGMP then dissociates from the active GC. Another cytosolic GTP is also converted into cGMP in presence of active GC. As a result, the cGMP concentration in the cytosol increases. The cGMPs then binds to the cation channel protein. Due to this cGMP binding, the cation channels become reopened. Through the reopened cGMP-gated cation channels, the extracellular sodium ions and calcium ions pass into the cytosol of rod cell. As a result, the VM returns to the pre-stimulus level. After bleached by the incident light, the active rhodopsin or RH with all transretinal will appear like this. The rhodopsin kinase, denoted as RK, then binds to the bleached active rhodopsin or RH. The rhodopsin kinase or RK then phosphorylates the bleached active rhodopsin or RH. The decreased calcium ion concentration and recoverin, denoted as RECOV, stimulate this RH phosphorylation. In the next step, the phosphorylated carboxyl terminus of active rhodopsin or RH binds to the arestrin, denoted as ARR. Due to this binding, the active rhodopsin or RH becomes inactive. The arestrin or ARR then slowly dissociates from the inactive rhodopsin or RH. The phosphorylated inactive rhodopsin or RH then undergoes dephosphorylation. All transretinal in the inactive rhodopsin or RH is replaced with 11-cis retinal. Now the inactive rhodopsin or RH with the 11-cis retinal is ready for another phototransduction cycle. Please like, subscribe and share.